Okay, I guess we have started to record. So welcome everyone to this week's AI seminar. We're very happy to have Dr. Rna Bernasconi. She's a postdoctoral researcher with Politecnico University of Milan, and also a visiting researcher at Politecnico University of Valencia. So her research areas are bioinformatics, databases, and data science, where she applies conceptual modeling, data integration, and semantic web technologies to biological and genomic data. So starting from a Ph.D. thesis on the modeling and the integration of data and the metadata of human genomic datasets, she has then extended her expertise to the fastly growing field of viral genomics, particularly relevant to since the COVID-19 pandemic outbreak. So she's active in the conceptual modeling and database communities with several paper presentations and the organization of tutorials and workshops. So today, Dr. Bernasconi is going to talk about data-driven SARS-CoV-2 understanding and hunting.
Thank you very much for the introduction, and thank you for giving me this opportunity to present this work to this very interesting seminar series. So the talk indeed speaks about data-driven SARS-CoV-2 understanding and hunting. As I've been already introduced, I will skip these slides, just saying that I work at Politecnico di Milano within the genomic computing group and inside the bioinformatics and data science lab. So my main interests are bioinformatics, databases, and data science methods. Today, I will speak about the most recent work, which is about viral genomics, and we have used viral sequence modeling, knowledge integration, search system, data analysis for trying and mitigate the effects of the COVID-19 crisis. Very briefly, I will start this talk in the context of the outbreak of the COVID-19 pandemic. The outline has an introduction about insights on data availability on the SARS-CoV-2 virus. Then I will provide a very brief viral genomics primer, as I think that the audience today may be very diverse, so someone may need some introduction in order to follow the second part of the talk. Then I will speak about the models that we use for SARS-CoV-2 data to understanding and actually organize the data. We will use some tools for understanding the data and some tools instead for hunting variants of the virus. And finally, I will conclude with some hints on how to get knowledge from literature on COVID-19. We'll conclude with the final remarks. Well, first, data availability. So, as we all know, COVID-19 pandemic was very disruptive, especially in the research world. We had a peak of interest towards this topic, as of course, previously, we only had the information about HIV virus, influenza, SARS-CoV-1, MERS, but indeed in 2020, we saw that there was a peak in the production of publications. This graph is showing the NIH PubMed database entries, which indeed in 2020 exploded. Another kind of data that exploded was the sequences of SARS-CoV-2, what we call genomes, the ones that are actually derived from swab tests that are collected in hospitals, in labs, and then sequenced by sequencing machines and technology and deposited to open databases. We started with a few hundreds in January 2020 from China. Then we reached 1 million about in March, April 2021, 6 million at the end of the year, and we today reached more than 13 million as of September 2022. So one interesting aspect when we look at this kind of data is the percentage of deposited sequences with respect to the number of COVID-19 cases in each country. Here you see plotted the deposited sequences versus the COVID-19 cases divided by 100 in orange. So those countries that have the same height basically sequence around 1% of the COVID-19 cases. Well, the USA case is a little bit more fortunate, as around 2.5 swab tests were actually sequenced. Italy did less than 1%. There are other countries that did much more, as United Kingdom, Germany, Denmark, Canada. Let's say that France and Italy were about 1%. Why is it interesting? Because of course, swab tests are just an indication of how the pandemic is going. But when you can observe sequences, you can actually observe much more fine-grained phenomena, evolution of the virus in a specific location, in temporal scale, in spatial scale, and of course, when very low percentages of COVID-19 cases are then matched to sequences, of course, some phenomena cannot be measured. Another aspect that we may want to look at is the difference between the collection date and the deposition date of a sequence. Indeed, what happened in year 2020 was that there was a very long delay between these two time points. So in other terms, a swab test was collected. So it was actually measuring, capturing something that had happened in that collection date, but more than 6-7 months had to pass before on open databases we could see the sequence matching that actual happening. So this was, of course, very bad for doing real-time analysis or even trying to capture how the virus was evolving in some countries. This luckily got very better in year 2021. As for example, I'm just highlighting Italy and USA, as I come from Italy and I'm giving this talk for you in California. So in Italy and US, more or less 40 days were required to deposit sequences in 2021, while in year 2022, luckily, we are down to 20 days, more or less. So when I speak about sequences, what do I really mean? Let's get into some small biology. So we all know the DNA that is enclosed in our cells. DNA is a double helix, double-stranded. It replicates, and then it transcribes into RNA. RNA is just one single strand made of several nucleotides, 4 kinds: adenine, cytosine, guanine, and uracil. 3 by 3, the nucleotides group and form amino acids. Many amino acids form proteins. RNA viruses are actually very common. So almost all the viruses that we mention every day are RNA. They are usually single-stranded, even if 2-stranded also exist, and they are made as their genetic material or ribonucleic acid. They include the common cold, influenza, SARS-CoV-1, MERS, dengue, Ebola, hepatitis, polio, and measles. Let's get into the details of the SARS-CoV-2. SARS-CoV-2 is actually very similar to the SARS-CoV-1, which caused the pandemic in 2002, 2003. It is formed by 4 structural proteins that confer the shape, the famous crown shape. Outside, we have the envelope, the membrane, the spike. Inside, we have the viral genome with the nucleocapsid. About 30 kilobases, so 30,000 letters form this genome. And all these letters all together can direct the synthesis of proteins. So here below, we see the proteins. The S1 is actually the spike, which is towards the right hand of the virus. And what happens is that along the different proteins, we have somehow mutations. These circles actually express one way in which we can express mutations. Mutations, what are they? They are errors during viral RNA duplication. So new virus every day are produced. They are similar to the original virus, but usually they're not exact copies because they have mutations. How do we encode mutations? Through triplets, which contain the reference nucleotide, the one that is supposed to be in the original virus, the coordinate from 0 to almost 30,000 coordinate on the genome, and the alternative nucleotide, which is in this new virus. We said that 3 by 3 nucleotides group and form amino acids. So if I change, for example, the A nucleotide in the middle of this triplet, I change it from A to G, I will then translate into a different amino acid. So from the aspartic acid, I will have the glycine. This is the way in which we actually express amino acid changes. In this case, we have a change in the spike protein, D614G. You might have heard about this one. It was quite famous at the beginning of the pandemic. And this is exactly the result of the first mutation that we're looking at. Viruses with mutations are called, and will be called in the rest of the talk, variants, also lineages. Variants could differ by a single or many mutations, and we can actually represent different variants within a phylogenetic tree, also called evolutionary tree. The evolutionary tree is, let's say, a diagram showing the evolutionary relationships among different sequences based on common or different mutations. You may have heard about the famous alpha variant, the one that started in UK at the end of 2020. When we speak about the alpha variant, we speak about the set of different amino acid changes that characterize this variant. Sometimes these effects, either alone or in groups or all together, have some effects, such as giving a higher infectivity to the virus, a higher transmissibility, or a higher severity of the disease. Same can be said about the Delta variant. You will see more changes here because the Delta variant came later, so the virus in that time accumulated more mutations. Which data models do we use for expressing this word of data? So we start from the sequence, which is the central unit of our interest and our computations. It has some information such as a nucleotide sequence, the strand, the length, the percentage of G and C nucleotides, the percentage of unknown nucleotides. It is also described by other information, biologically speaking, by the information of the host sample, so the host organism, a person, or also an animal that hosted the virus, that was infected by the virus. It is matched by information about the collection date, where the virus was isolated, if from the throat or from nasopharyngeal swab. Some other information about the geography of the swab. Then we have some information about the virus, because of course this model can be used for also other viruses in addition to SARS-CoV-2. Then we have information about the sequencing project, so the lab and the project that actually made it possible to sequence the biological material. We have information about the technology, so the sequencing platform, the assembly method, the coverage. And finally, we have the most interesting part, which has to do with mutations. As we said, we have nucleotide mutations, but also amino acid mutations that belong to a specific annotation or protein. Mutations also have effects. Effects could be represented through a simple taxonomy that I'm just showing a part of it. We have 4 categories. Effects could could be on the viral kinetics and dynamics of the virus, or on epidemiology, for example, viral transmission, infectivity, which are actually not the same thing, as viral transmission is the ability of the virus to pass from one host to the other, while infectivity is the ability of the virus to establish an infection within the host. We have disease severity, fatality rate. We have effects that have to do with diagnosis and prevention. We also have immunology. We have immunological effects, which basically have to do with the sensitivity of the virus to some treatments, as vaccines, as monoclonal antibodies, as the sera of convalescent patients. Also, we have the binding affinity to host receptor, which is the ability of the virus, this part of spike indeed, to attach to the ACE2 or any other host receptor. In the case of humans, it's this enzyme, which confers it the possibility to enter the host cell. Another model, which is more abstract and it actually includes the one that I just described here in the top right corner, is the CoV-2K model. CoV-2K model was devised with the purpose of putting together data and knowledge. On the right side, we have data, which is the big data. So here we have the 13 millions of sequences. We have epitopes, which are parts of the virus that are studied and used for treatments. On the left side, instead, we have what is established knowledge about the virus. We have the variants with their different names provided by different organizations, characterizations, which are the sets of mutations that characterize a variant. They could be either amino acids or nucleotides. They could stand in a specific region or annotation of the virus within proteins. They change molecules. So within a change, I have the information about changing from, as we said before, for example, aspartic acid into glycine. So I'm changing all the properties of that molecule. It could be hydrophobicity, it could be a change of polarity, a change of size of the molecule. And then we have the most interesting part, which is the effects that are could be caused by either one change, a group of change, or an entire variant. Effects have a matching evidence, which could be a paper that has been published and explains about that effect. And just to give you a broader vision, I would like to point that today I'm speaking about viral sequences, but of course there is a whole world behind that, because a viral sequence is hosted by a patient, which will have connected information about the phenotype, so the clinical information, and then the genotype, so the genomic information. Unfortunately, this link is not available in open datasets, so we're not going to discuss about it, but we're actually working with small hospitals in Italy towards this direction. Databases and tools. What did we do with these tools? The VCM and the CoV-2K were actually used to design databases that store a lot of sequences, actually millions now, taken from the most known data sources. We had to actually build two different databases, as the GISAID source asked us to keep data separate from the others. And then, thanks to building these two databases, we then built many other tools that I'm going to describe next. Our Etl pipeline can be described as an ETL pipeline that stores, extracts the information, transforms it, and stores it into a Postgre database, while for the knowledge part, instead, we use a MongoDB database with some harmonization steps as reconciliation, duplicate removal, cleaning, and coordinate translation, with the purpose of organizing everything within a RESTful API that is open to everybody. So very briefly, I'm going to list the 6 tools that then will be described more in depth. We have VirusSurf, which is a search engine. EpiSurf that puts together the viral sequences extracted with VirusSurf, and epitopes to test the stability of epitopes, which are used in vaccine design. VirusViz is a visualizer. We will see some details later. Viroplast instead allows to aggregate groups of sequences to observe the differences of mutations. Variant Hunter, as the name says, allows to observe variant trends and then identify novel emerging variants. And finally, Convolution is not yet a tool, it's just a method that allows to recognize variants at early stages and provide warnings. I divided these tools in those that allow to understand the domain and those that instead hunt for new variants. So for understanding, VirusSurf is just a search engine. We translated the ER model, the entity relationship model that I show, into a search interface that allows to compose conjunctive queries over metadata and then combine them with patterns built on the variants. So I will be able to, for example, select sequences from this country and that period of time and with a specific pattern, with this and or that mutation. And I will obtain, as a result, the matching sequences that respect the filters I set. Epitopes. What are epitopes? They are short sequences of amino acids inside a virus protein. So you can observe it in the small figure. This is a virus in purple. There are some small sequences in yellow called epitopes that are recognized by our antibodies and therefore can provoke immune response. These epitopes are stored and curated in the immune epitope database, actually curated close to you at La Jolla. Now we have 1 million epitopes, 6,000 are for SARS-CoV-2. One curiosity about the designer of Pfizer and Moderna vaccines, apparently they use all available epitopes on the spike protein. And why are we interested in studying mutations on epitopes? Because they actually might compromise the recognition of epitopes from the immune system. So this is why we build a system that allows to intersect what is sequences, so we take them from these 3 sources, and what is an epitope. So basically, the users can search for a specific population, for example, all the sequences extracted and collected in California, and check which are the epitopes that are most impacted by the mutations in the sequences from California. So this is an example of a search that I may do on the system. I may search for sequences in UK, the early Delta variants. So I extracted 150,000 sequences. You can observe these black bars are counting how many sequences out of the total have a mutation in these points. So on the x-axis, we have the spike protein. And actually, we see that in this position, all the sequences have some mutations, because of course, these are 2 mutations that are typical of the Delta variant. The point is that for this specific epitope that we extracted from the system, this epitope is actually very impacted by these 2 mutations. So the idea is to suggest that all the epitopes that are highly impacted by the mutations in a specific sequence population should be checked, and also the assays that use them should be checked for if their functioning is impacted or not. And here we have VirusViz, which is our visualizer. This visualizer allows to build the tracks where we count the bars, count how many sequences have a mutation in that specific position on the coordinates of the virus. This was a very old example that I still like very much, because it shows even if with very small samples, because here we have about 50 sequences in each track, it shows how Italians returning from vacations back in 2020 came back with many mutations, because in the first track you have the profile, the mutational profile of before the summer, during the summer, and after the summer. And you can observe how the last track is much more mutated, as of course people came back from traveling around all Europe, and they had accumulated more mutations. On the same line, we have this example about the A22v mutation, which apparently came from Spain during the summer. Indeed, you can observe that during summer and after summer in Spain, we went from 46 to 70% of this mutation, while in Italy it was absolutely absent before the summer, and then instead it came to 39% after the summer. Another thing that we can observe with virus bits is the growth, the initial growth of variants. This is what we found for the alpha. We all received the warning about alpha around December 2020. Here you can see that already in the second half of October, then this tracks group sequences 2 weeks by 2 weeks. So I have the second half of October, the 1st of November, the 2nd of November, the 1st of December. You can see how the blue and orange rectangles are highlighting some little soldiers that are growing together from 0 to 17%. So already at this point, we could have said something about alpha. And the same, I also ran the query for the Epsilon variant, which you may have heard because it was supposed to have born in California. For this one, the first alert from the scientific community was in a letter of February 11, 2021, but indeed already in November, December of 2020, we saw these 3 mutations that were from 12% and they reached 61% in February. Then I would like to show the fourth system, which is called Viroplast. This allows to test some hypotheses. So let's suppose that we want to study the early evolution of Delta, and we ask if there are mutations in India, which was a likely place of origin, that are not observed in the rest of Asia, or vice versa. So in order to perform this search, we can run a spatial analysis on Viroplast, checking the B16172, which are the Delta sequences, checking India versus Asia, meaning Asia excluded India. And then we will extract a meaningful period of time, which is from mid-2020 to mid-2021. What we obtain is a bar plot that is showing how many sequences in India, which is our target, and how many in Asia, which is our background, which of course is a little bit later, some months later. What we can do with the spatial analysis is obtain a table of this kind that will pinpoint that for 5 mutations, they were not so present in India, with percentages around 30%, with the exception of this one, which was 33%, but they are fixed, we say fixed, they reach fixation after 90, with more than 90% outside of Asia. We want then to compare this with a temporal analysis. So we ran a temporal analysis on the 3 most interesting months from May to July 2021. On the left, we have India, on the right, Asia. Here we have what the so-called heat maps. So we will see a blue color when the percentage of presence of that mutation is low, and a red color when it's very high. So here we observe that there are actually 5 mutations that were not so present in India, but reach much higher percentages in Asia. And here we come with the tools for hunting variants. So as the name says, Variant Hunter is a tool that allows to analyze frequencies of amino acid mutations, to observe interesting variant trends, or to identify novel emerging variants. The most important number that we use in Variant Hunter is the frequency of mutation in a specific week. We compute it as the number of sequences exhibiting the mutation during the week divided by the sequences collected during that week. We use 4 of these frequencies. So we observe 4 different points, 4 weeks, and we fit a linear model on these 4 data points. Then we interpret the slope of this regression line as the one that indicates how fast the mutation is growing. Thanks to this mechanism, we were able to pinpoint many interesting facts. For example, we observed the early spread of Delta in the UK during April-May 2021. You see there is a bundle of lines that are decreasing here. This is a mutation that is considered as the most characteristic of alpha. And here you see a bundle of other lines with the most characteristic of Delta. So of course, here we can see the trend, which is that Delta is rapidly becoming predominant over alpha. Another observation can be made about the spread of Omicron during the concomitant displacement of Delta. This happened in around December 2021, January 2022. Here there is a comparison between what was happening in Europe and USA. Of course, this is what we observe through the data. So in this case, in the US, you can see that the signal appears less accurate, probably because there were less sequences at that time. But more or less, the behavior that we can observe is similar. So there is a bundle of lines decreasing. Actually, we can observe it also using the heat map from red to blue, and a bundle of sequences, of lines that is increasing, so going from blue to red. Another use of Variant Hunter, so it has actually 2 modes. The second mode is used to identify novel emerging variants. So our starting point was a hint from this forum, which is called CoV Lineages, Fungal Designation Issues. And this is the most used between phylogeneticists to study the evolution and to study and actually discuss about the evolution of the virus. They were talking in early April 2022 about the possible sublineage of this A22v variant, which is indeed the Omicron 2 sublineage 12. What we are able to do with Variant Hunter is to observe that there was actually a lineage BA2-12-2, so another sublineage that was detected in March 2022. So 1 month earlier, we were already able to observe that there was a variant, namely the L452Q, that was going in just 4 weeks from 0 to almost 20%. This could be observed more directly through the heat map that shows that indeed this mutation has a very different behavior as when compared to the others. And we can also integrate this analysis with a diffusion odd ratio, where it is clear that this mutation is growing. And finally, the 6th tool that I wanted to talk to you about is actually a method that is called Convolution. We're working towards making this a tool, so to reproduce the analysis that we made at this time. This is an analysis of the time series of amino acid changes. The focus here is on the co-occurrence of mutations that we call amino acid changes, as we want to look to the mutations that occur at the protein level. The idea is that a variant can be identified by observing the dynamics of its amino acid changes. The dynamics are frequencies. They are the weekly frequencies in a geolocation. Then different changes could indicate the birth of a variant when their time series are similar and they are all growing. Let me provide you some visual support to this intuition. Here we see a snapshot of what was happening in the UK from May 2020 to March 2021. All the lines here are the weekly prevalences, weekly frequencies of each mutation. So, for example, if we see the spike D614G, it was already very broadly present. So in May 2020, it was already at 90%, and then basically was present in all the sequences that were collected after. Then we have the NR203K, which instead had a more sinusoid behavior. We have other ones that never reached more than 10%, so are not really interesting. And instead, what is really interesting is this part. If you see here, at this point, we have a bundle of mutations that are actually growing together. This is the interesting kind of behavior that we're looking for. So we wanted to build a method to recognize early enough the growth of variants. And this indeed turned out to be the UK variant. How does the data analysis method work? It is a time series clustering. So we first ask if temporal patterns occur in a coherent manner. Then we ask which clusters could signal the emergence of a variant. So out of these three that I captured, I only kept the first one because I'm trying to give early warnings. So I just want to retain clusters that are sufficiently new with respect to the others that I observe. And finally, we compare the clusters that we identified with the known lineages. So we check the compositional match between clusters and lineages that are known a posteriori. And this analysis allowed us to achieve very good results. In other terms, what we did was run our method for the 8 WHO named variant, alpha, beta, gamma, delta, and so on and so forth. We ran it towards them. And what we obtained was a hit date. So the time when our method would have thrown an alert, an early warning. And then we compared that to the first communication that was given from health authorities of the countries of origin of that variant. What we were able to do in 5 cases out of 8 was pretty impressive because we were able to throw an alert at least 2 weeks earlier of when compared to the health authorities communication. In 2 cases, we came at about the same time with our alert, and just in 1 case, we arrived 2 weeks later, probably because of the lack of data. Because, of course, everything that we can observe here strongly depends on the availability, on the timely availability of data from the country where the variant is growing. So as outcomes of this project, we could derive that the emergence of variants can be traced through purely data-driven methods, and that an early warning system could rely exclusively on deposited sequences. Finally, I would like to put your attention on the fact that in this model that I showed at the beginning, we're able to fill information about sequences, about epitopes, about variants, about mutations But indeed, how did we fill this information? How did we fill our databases with effects? It is an incredibly cumbersome work that was done up to some point manually by our expert in biology. So we were trying to find a way to do this automatically or in a more aided way. So first of all, we looked at the literature. Literature is enormous for COVID-19. Already during 2020, there was an estimation of the growth of this amount of papers going to 200,000 papers, but we reach actually more than 1 million. Indeed, everything is contained in the Core-19 dataset, the COVID-19 Open Research dataset that has been created by the Allen Institute for AI, maintained by Semantic Scholar, and for supporting text mining and NLP research. It was launched in March 13, 2020, with about 40K articles. In the final version, which is of last June, it was indexing more than 1 million papers. So we extracted only the part that has an abstract, not only other kind of metadata, and we analyzed all this corpus. We were able to find through a basic k-means algorithm 5 different clusters, one about epidemiology, one about biology, then therapy and treatment, psychology, and society and environment. We took a special interest in the ones about epidemiology and biology, most of all biology, and then we tried to understand how to automatize or to support the extraction of information about variant effects. We came up with this idea. So actually, what we wanted to do was to extract information such as you can see in the first abstract here, increased case fatality rate correlated strongly with the proportion of viruses bearing G614. We wanted to transform this kind of information into structured information such as spike G614G leads to higher fatality rate, or something like in the second abstract, variants, blah, 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 became resistant to some neutralizing antibodies. We wanted to extract that the spike A47V led to lower sensitivity to neutralizing monoclonal antibodies. How could we support this? We came up with the idea of an interface that allows expert users to use a keyword search to find papers that are relevant to the aspects that are interesting for them. So they get this paper list, and then one by one, each paper is analyzed in this interface. There is actually a transformer-based GPT-2 model behind, which is trying to predict from this abstract all the annotations that are made of a mutation, an effect, and a level. Here, for example, the model was able to understand the mutation, spike and 501Y, the effect, binding to host receptor. It didn't find correctly the level. Indeed, the information about the level was here. So an expert user could read this information and actually input it by hand in the system. The system takes this information and uses it for retraining the model in an active learning process. We're using saliency maps here to highlight what information is actually used by the model to make the prediction. And we really hope that this system will speed up the filling of this kind of information for our knowledge base. The very last thing that we are that I was going to tell you about is what we are doing with Core-19 in terms of expressing the co-occurrences between concepts. We're building a co-occurrence network. We take abstracts we match them with the concepts from the Unified Medical Language System. So our entities are concepts and our relationships are co-occurrences between these concepts. So if I have these 4 concepts within the same abstract, then I will make a +1 in the weight of these relationships. How do we use this network? The idea is that a researcher could express an original hypothesis, that she would be able to express it in the form of a graphical abstract. For us, graphical abstracts are subgraphs. So in this case, as it became unreadable, I made a subgraph saying that SARS-CoV-2 is connected to the enzyme AC2, is connected to angiotensin, and connected to vascular permeability and procoagulant state. What do we do with this? We look for it in the co-occurrences graph. It is on a Neo4j database. We run this graph mining and we obtain an explained graphical abstract. So the parts of the graphical abstract that were not directly found on the graph could be found using different paths on the graph. So this explained graphical abstract also corresponds to a long list of papers that are ranked by a number of covered explained relationships and the NMPMI, the mutual information between concepts. So we really hope that this could help researchers that have an hypothesis and are looking for a relevant bibliography to study that hypothesis. So in summary, I started from the outbreak of the COVID-19 pandemic. I talked to you about data models, tools that are used either for understanding the data or for hunting variants, and how we plan to enrich, augment our knowledge in the database from using COVID-19 literature. In summary, I hope I've been successful in showing that the data-driven approach can analyze SARS-CoV-2 virus characteristics and can gain insights on their evolution. As general objectives, we have developed methods and tools that are dedicated to generic users. Anyone can use them. I showed some links, you can try them, but are also needed for performing our own analysis, as we did for the variant's birth identification. More in general, our high-level goals are that of making the pandemic explainable by exposing and connecting viral data with knowledge. And we also try to define and resolve new computational challenges in viral genomics, taking advantages of data science methods. And here I can conclude that here is a list of relevant publications of which I've talked about, and special thanks to all the group at Politecnico di Milano that has been collaborating with all this work. And thank you very much. If you have any curiosity and questions, I'm here to answer.
Okay, thanks, Anna, for this fantastic talk and the introduction to lots of new technologies and tools. I see that in the chat, Abigail has asked 2 questions. Abigail, do you want to ask the questions by yourself?
Sure, yeah. So the first one was just a clarifying question, and thank you so much for your talk. It was fantastic and educational. So thank you for breaking things down for us. The first question was just, I guess basically, could you talk more about epitopes, and is that information generally available? Are you using it in any of the methods that you're, these large scale methods that you're applying for early detection, or is that kind of like a more secondary piece of data?
Yes, thank you for your question. Of course, so I probably should have been clear about what epitope data is. So mainly our focus has been on sequence data. Okay, sequences are deposited on open data databases. Epitopes instead are a different kind of data because they are just fragments of the sequences, and they are fragments of the reference sequences. So they are studied on the viruses as they should be, so not the ones that are mutated. So they took the sequence from Wuhan, the first from China, and on that they tried to understand which parts could be useful for design vaccines, design therapies, drugs, assays to understand actually the COVID tests that we use every day are all based on epitopes. In principle, epitopes should be parts that are quite stable, so parts that don't change so much, so you can keep using them for quite a long time. Unfortunately, as SARS-CoV-2 kept mutating as we expected, epitopes may become obsolete somehow. So that is why we built a system, EpiSurf, to put together the information about sequence and epitopes. Epitopes are available in the IEDB database. That is a database that is manually curated by immunologists. It was, as I said, it was created in La Jolla. I don't remember exactly the name of the lab that is running it, but he's been there for many years now and collects epitopes from many different virus species. I don't know if I covered your doubt.
That was really helpful. I have a better picture of how that information fits in and when it's used, when and where it's used. Great, so thank you so much. And then the second question I had was with the methods for early warning, basically emergence of new variants. And so here in Los Angeles, we have a pretty extensive sequencing program. The county health department runs it, and we sequence around 10% of cases. Now, many fewer cases are actually going to sequence, but there's random sampling programs and focused on specific populations of interest, like care facilities and that kind of thing. Mm-hmm. I was wondering, you know, the methods that you've developed for early warning, the nice time series clustering methods, do you think these would apply well in a more localized context? Again, you know, provided that sufficient kind of sequence density?
Yes, thank you. Thank you for this point. Actually, my answer is absolutely yes. And you already answered yourself, provided that enough data is available. Indeed, we are trying to do this in Lombardy, which is the region of Milan where I live. The genomic surveillance here is not as rich as it could be, so we don't have enough data to see everything. Also, they prefer not to sequence, but rather to use a real-time PCR, which only captures some mutations on the spike protein, which is quite useful to catch information such as, okay, I'm getting a sequence, I'm getting a swab test that doesn't have the mutations that I usually see in the sequence in these days. So that could mean that a new variant is coming. But indeed, that kind of information is not enough to run our kind of method. Right. I would say with California, we had actually good results, because at the time when we applied our method, we were testing it for the Epsilon variant. So in that case, the data was enough, and I expect now to be even better, because I expect the facilities to have improved and so on and so forth. Yeah. So yes, I mean, it would be very, very nice to try it even on smaller local data, even because I expect not all the local data to be made available on open databases. As it always happens, there is, you know, some difference between what is kept inside and what is provided for the worldwide.
Yeah, that's definitely the case here. So great, thank you so much.
Thank you.
Exciting.
Thank you for your interest.
Yeah.
Should I go on with the question from Craig?
I can go ahead and just ask my question.
Okay. Yeah.
So I think the mapping from the variants to their effects is actually really interesting. And so you talked a little bit about, you know, getting that information out of the papers, and it sounded like you were doing that, the extraction of the data from the papers manually, or is there some automatic method for actually extracting it? And if not, have you looked at how to automatically extract that kind of information from the papers?
Yeah. So indeed, up to some months ago, we were doing it manually. Under double quotes, because some websites provide some aggregating information about this. For example, the COG UK genomic surveillance system in the UK is providing some information. Other websites are providing information about variants. So we basically had an expert, a domain expert, that was going through all these different resources to find this kind of information. Then we started trying to extract this automatically. And this is where we came up with the COV effect system, which is the one I showed, that it's actually something that is supporting the expert in annotating data. And meanwhile, it is accumulating training data for our transformer-based model. So this was our best solution for now, because we try to make this completely automatically, but there is no training data for this kind of problem. It is very specific and it requires a domain knowledge which is really intense. So unfortunately, without training data, and we tried with pre-trained models that are, you know, for biomedical domains, but this is just so specific, and that we couldn't achieve anything usable. So that's why we created this system. It's actually going pretty well. We were reaching good performances, and hopefully it will be out so that maybe the community will help accumulating more training data. So this is
Great, thank you. That's really, really nice talk. Thanks very much.
Thank you. Thank you.
Any other audience? Any other question from the audience? Yeah, actually, I have a question, but that's just out of curiosity, because, you know, I'm a natural language processing and machine learning person. So I'm actually wondering whether in your tool or sets of technology, you would be thinking about supporting any dense representation-based retrieval of the sequence data, because my understanding is that genomic data and proteomic data, they can accumulate into a very large amount, right? So if we go to the string database, even we just want to do a one pass scan, it will take multiple weeks even. But if, let's say, we can convert some of them into embedding using maybe proteomic language models, then everything will be in the vector space, and that will become easy to index and search.
Yes, we haven't explored this kind of models yet, because as you may have grasped from the presentation, the work on understanding information from the text is very new, is the most recent result that we have. We don't even have any publications yet. So let's say up to slide 45, it was all quite established and published, whereas the last 5 slides were what we are trying to do in these last days. Indeed, we absolutely need to go deeper in understanding what models can be used more successfully to grasp knowledge from this big data, big corpus of papers, which you may know about, COVID-19. There is so much to extract from them. We started from a very specific problem, which was the information of that connects a mutation to a specific change effect in the behavior of the virus. So now we're trying to understand if a mutation provides a higher transmissibility, higher disease severity to the virus. So for this specific kind of task, we found that the translation-based models are quite appropriate, more than some other named entity recognition that we did before. But of course, we're open to other ideas and other collaboration as well.
Cool, thanks, Anna. So I guess we have passed 12 already, so this could conclude our seminar today. Thanks once again for this fantastic talk, and look forward to the follow-up discussions with you in the one-on-one meetings.
Thank you to everyone for the interest. Bye-bye.